I put this graphic together on the aluminum toxic effects, all from scientific literature. With everything coming out about vaccines, this may be helpful to see several mechanisms in which aluminum can affect our biochemical processes. So this is a lot, but definitely not all inclusive because I haven't reviewed all the aluminum toxicity studies. I'll do a quick run through of what you are seeing. Arrows are pointing to aluminum, and that's how we get exposed to it. It is an abundant element, so we're going to be exposed to some, but I focus on the two main pathways of how it gets into our bloodstream. Through oral ingestion with what we eat and drink, and then vaccines into the muscle, where almost 100% goes into the bloodstream and binds to transferrin and citrate, and gets carried to our organs and tissues to accumulate, especially if we're not able to detox efficiently. Then you see several of the main organs' aluminum effects and what goes on as biochemical processes from there that cause oxidative stress, mitochondrial dysfunction, neurotransmitter imbalances, microbiotic changes in intestinal and blood-brain permeability that, again, worsen the effects from toxins, global hypomethylation, neuronal injury, and neurodegeneration. If you would like a copy of this, comment chart. And if it would be helpful to dive into each of these sections more to explain these mechanisms, respond to the poll below. We're going to dive into different sections of my aluminum toxic effects graphic to explain what you're seeing. It's going to take several videos, but worth it to see how aluminum impacts our bodies and health. First, how are we exposed to aluminum? How does it get into our tissues? The most common methods of entry are by mouth with what we eat and drink, and then vaccines where it's included as an adjuvant. Because aluminum is the third most abundant metal in Earth's crust, we're going to have some exposure, but how we're being exposed matters, and how much. So if you take an acid, for example, that can drastically increase your daily exposure and absorption because of the high amounts of aluminum. It is primarily excreted by the kidneys, but if you have issues with your kidneys, well then you can't excrete it efficiently. And this graphic isn't accounting for that or any of those factors that influence the risk of aluminum exposure or an infant with immature systems. Inhalation is another method, so exposure in an industrial work environment. And then we get to vaccines, where most are intramuscular injections. Almost 100% is directly entering the bloodstream. It may slowly release over time from that deposit in the muscle, but it is still then directly going into the blood. And once in the blood, about 90% is bound to transferrin. That can take it to the brain and other tissues. It can also bind to citrate or passively diffuse into the brain. So there are actually 3 pathways on how it can get directly to the brain and easily enter to accumulate as well as in other tissues. The main difference with oral intake versus IV or IM is that the oral intake isn't going directly into the blood. A majority is being removed, although there are a lot of factors that can affect how much you're absorbing. Okay, so the amount in vaccines and milligrams compared to the amount in milligrams that you're taking in by food are not the same. So follow me for part 2, where we'll continue to dissect this graphic, and please drop any questions as we go in the comments. You can also comment aluminum if you haven't received a copy of the graphic to follow along. Now we're going to look at the effects aluminum has on the intestine, where aluminum goes after oral ingestion, focusing on this area here. What is aluminum doing at a cellular level? Multiple things, but we're going to talk about some of the big ones. First, aluminum causes oxidative stress. The study shows that cellular oxygen stress levels, specifically superoxide production, when treated with aluminum was around 38-fold higher than the control group, which you can see in figure 2 with the red fluorescence being superoxide production. You don't really see any in the control group, and then in the high aluminum concentration, it's very bright. Lower concentrations of aluminum were used because the higher concentrations had significantly lower cell viability, meaning dead cells, and they needed healthy living cells to do the studies. So that says a lot right there. The higher aluminum concentration had more dead cells. It also showed that in human colorectal epithelial cells, or HT cells, aluminum activated inflammatory cytokines such as tumor necrosis factor alpha, interleukin 1 beta, and interleukin 6 cytokines, which we can see here. The higher the aluminum concentration, the higher the inflammatory cytokines. It also down-regulated tight junction proteins, claudin and occludin. So these are what you can think of as keeping the integrity of the intestinal barrier, or everything compact rather than loose. Aluminum induced histological alterations where you see blunting of villi, abscesses that typically form in response to inflammation, and an infiltration or leaking. Also, look at these pictures of mouse colons. The weight and length of the colon decreases with higher aluminum exposure. So all of these effects, along with generation of oxidative stress, cause epithelial barrier dysfunction or leaky gut, where substances can pass through more easily. substances like bacteria, toxins, undigested food particles that shouldn't be getting through your intestinal barrier, and then they get into the blood and cause systemic effects. What we just talked about covers this section of oxidative stress, downregulation of tight junction proteins, increases in intestinal permeability, and immune response with activation of inflammatory cytokines. But I'll go more into the immune response in part 3, so follow me to learn more. Still focusing on the effects aluminum has on the intestine. We previously talked about the oxidative stress area and that aluminum has been shown to activate inflammatory cytokines that trigger a response. But aluminum does more than that. The study showed that aluminum reduced the production of IgM and IgG and activation of B cells, CD4T, and CD8 T cells. Simplified, it suppressed adaptive immunity. Aluminum had a direct action on B1 cells, inducing B1 cells to produce interleukin 10, an immune suppressive cytokine. And just to review, adaptive immunity is your memory immunity, where you produce antibodies to pathogens and then also get rid of unwanted cells like cancer cells. Also, why aluminum is used as an adjuvant in vaccines to boost your response, as aluminum potentially enhances the antigen presentation. Aluminum, though, here reduced production of antibodies and impaired activation of B cells by activating the JAK-STAT pathway and increasing interleukin 10. If we think of this in terms of why aluminum is used in vaccines, to boost the response to make more antibodies to recognize the pathogen, but here we are showing a suppression of immune response, which completely invalidates the entire purpose of aluminum being in vaccines in the first place. Which brings me to the question of, if we had this evidence that aluminum may suppress our immune systems, why wasn't this study replicated in humans to further evaluate this to confirm if it's truly warranted to boost the immune response when aluminum is a well-known neurotoxin and we're giving it to newborns dose after dose? Question I just don't understand how we've gone so many years without it not being addressed. Anyway, back to my graphic. We've talked about the immune pathway, so follow me for more in part 4 to discuss the effects on our microbiota.